All right, good afternoon, Cell Bio. How is everyone doing? Hopefully you're all doing okay. So just a couple of things. I've only run a few of these Zoom sessions so far, so it's still a little bit new to me. I'm quickly trying to learn the ropes to hopefully get this down so that it'll be better every time we do it, when we do in fact meet this way. One quick thing to start off, I have you all on mute. If you do have a question, use the raise hand function. I should be able to really quickly see that when the raise hand function comes up, and then I'll call on you. You can unmute yourself, and then you can ask a question. So, oh goodness. Okay, so you can't hear me. Let's see. Okay, let me actually take one step back. Can a few of you use the chat function and let me know if you can hear me? Yes, okay. So then there was one person who said they couldn't hear me. That might be specifically on your end. Clarissa, that might be a problem on your end. I'm glad you mentioned it, but again, this is still all new to me, so I'm still going through the ropes trying to learn everything. Okay, so seems like a few of you did in fact type in with the chat function and you can hear me, so that's good. So again, just really quickly, as we go through the information, I will pause multiple times and often so that there is time for questions. But again, if you use that raise hand function, that should be able to allow me to see that you have a question, and then I can pause and ask that question or answer that question. Really quickly, if you are still having trouble, let's see, I probably should message her if she can't hear me. She can't hear me telling her what to do. Let me see if I can message her really quickly and let her know. Give me just 1 second. How about I close my emails so that we're not hearing dings constantly as well. Okay, so what I actually wanted to start off with is first to let you know that I am recording the Zoom session. So a few of you did email me with questions. Will I post the Zoom session afterwards? And the answer is absolutely yes. I'm hoping though that when we do interact this way via Zoom, that you will have the time to come and participate, because this is our chance to really interact as close to face as we possibly can. And sometimes questions might come up during the Zoom session that we can answer sort of right as the time the question comes about. Let me admit a few more last minute people coming in. All right, so I actually, before we start with any lecture material, I want to jump on and just kind of cover the syllabus in a little more detail. Is everyone seeing the syllabus? Oh, I'm muted again somehow. Hold on. Okay, so a few of you have answered, you can in fact see the syllabus. I just want to make sure that you can in fact see that. All right, so just quickly then, what I wanted to make sure that you're aware of, are things that I did already post in an email previously, in that my office hours will stay the same. However, I'm not going to hold open Zoom sessions during my office hours. What I plan to do is, if you have an individual question that you would like to ask me, then I definitely will set up an individual Zoom session for you, rather than me trying to sit and wait for people just to show up for office hours. I would rather do that on a one-to-one basis. Obviously, you can still continually email me. I'm checking my email often and regularly, and hopefully being able to get back to those questions when those questions do in fact arise. Okay, so questions about how office hours will be handled. Again, if you have a question, just use the raise hand function, and then we'll go from there. Okay, so scrolling down just a little bit, what I did highlight was some of the changes that have occurred in the syllabus with this transition to our new way of teaching for the rest of the semester. And that is, we will have an exam 3. However, that exam 3 will only be worth 50 points rather than 100 points. I decided to do it this way just because we've lost about 2 weeks of course time or class time because of the transition that needed to occur. And so I've had to pare down some of the material, and rather than have one large exam at the very end, I decided it might be better to have sort of a little snippet of an exam, which will actually happen after this week. So we still have this week's material, today's lecture, and then the lecture that I'll post on Thursday for you. And that material will be what will be covered on this exam. Now, the exam will be available to you via Blackboard, and it'll be my first time trying a Blackboard exam, so there too we'll have to try and just do the best that we can with giving that a try. The quizzes, we've dropped those down in number, just a few, only because, again, we lost 2 weeks of time, and so we don't physically have the time to have as many quizzes as I had originally anticipated. Another large change was that we are not going to write a full-blown research report. What you will be doing is writing a lab report, a mini report, and that mini report will be worth 25 points. And I am going to post some information about that today. There was a little bit of information in the lab handout. Those of you who have already completed the lab are aware of that, but there's a little information there as well. And then lastly, the number of lab assignment points is going to be a little bit less, just because we've lost a little bit of time there as well. So any questions about the changes in the point system or the grade distribution overall? No questions? Okay. So just scrolling down a little more then. If you have had a chance to take a look at the syllabus, you can see the adjusted or the changes that have been made. This week is the fermentation lab, modified, updated fermentation lab. Next week will still be spring break, so we'll still hold that as spring break. And then the last 3 labs are listed there. I don't have them posted yet. I probably will be working through spring break to try and get all of that information ready for you and available to you overall. Okay, and then scrolling down just a little bit more with the topics that we'll be covering. As you can see, we had a holiday last week. I did post the respiration lecture voiceover last week. Today we will review all of respiration and add a little bit of fermentation. And then on Thursday, I will have another voiceover dealing with photosynthesis. So this exam that's coming up, the exam number 3, will focus on respiration, fermentation, and photosynthesis overall. and that will be available after Thursday. I will have it open for a while so that you don't have just one specific time point to get that accomplished. I will have it open for a while, and then we will continue on after spring break. And again, it'll be a combination of voiceovers and Zoom sessions. So questions about the syllabus overall? No questions? How about there we go. I just wanted to make sure that this is working. Amrit, go ahead and unmute and ask your question.
Okay, so for the midterm 3, so
Yes.
it's gonna be available for like 2 days, or like is this gonna be available for 1 day, and at whatever time you wanna do the midterm, you can take it?
Yeah, it'll be open for a few days. Now I haven't decided how many days, but it'll be open so that you have some flexibility. Again, I realize everybody has sort of just crazy schedule changes going on. You know, we all have different things going on at home. So I'm not going to tie you to, it has to be the specific time for that exam. I will leave it open for a few days, but I just haven't decided how many days yet. And then once you do start it though, you do need to complete it in that time frame that you started.
Okay, thank you.
You're welcome. Okay, any other questions about sort of logistics of how we're going to try and make this run through the end of the semester? No more questions? I do see a question, will the exam be timed? And I think I just answered that, but just to clarify, it will be timed. So once you start the exam, you can't close it and open it again. You have to finish it in one sitting. It will probably be a longer sitting than our normal class time, but that too, I haven't quite figured out the details yet. I'm going to start talking to some of my colleagues who have done exams via Blackboard and just kind of get a consensus of what does seem to work and what doesn't seem to work. We too, on our end and the faculty end, are just trying different things, and as we try them, we find out what does work, what doesn't work, and we actually do share information. So I will probably gather a little bit of information from my colleagues to see what a good time frame is for how long the exam will be open. Okay, so hopefully that answers your question, Angel. Okay, and then there was another question about if it will be on Thursday. So again, it won't be specifically on Thursday. It will be posted after Thursday, but it'll be open for a few days. And again, I'm not sure what days those are, but I'll have it open for a significant amount of days so that you'll have an opportunity to address the exam. Okay, so any other questions about the course? Again, how we're going to try and run it overall? No. Okay, so let me transition here.
Dr.
Yes.
I actually had a question.
Okay.
Can you kind of elaborate more on how the labs are going to work from here on out?
I will try. I'll do my best there. My plan is to either have material that is prepared ahead of time. The last few labs are not necessarily, for us it worked out that they're not necessarily ones that we actually have to be there. So I'll either have similar options like a YouTube video that you might watch. For one of them, I might actually compile a video on my own. But so there might be something that you need to watch in order to sort of participate, if you want to call it that, in the lab. And then there will be some activity or exercise associated with it. Does that answer the question?
Yes, thank you.
Okay, great. All right, so let me go ahead then and move over to the slides for today. All right. Okay, so what I want to do is go ahead and hopefully you have had a chance to go through and listen to the first lecture I posted on cellular respiration. This should be now a review. So my intent here is not to cover all of the detail that I covered previously, but just kind of reinforce the material that we have talked about with the previous lecture overall. So the first thing that I want to review is just in general what cellular respiration really is. So cellular respiration is defined as a catabolic energy yielding pathway. It is the #1 pathway that allows for the production of ATP. In this particular pathway, organic compounds, food or fuel sources are utilized with the presence of oxygen to then produce carbon dioxide, water, and energy. And again, the important thing here is that these are catabolic reactions. And again, catabolic reactions are those where large molecules are broken down into smaller molecules. And in that process of breaking down large molecules, which store a great deal of energy, in breaking them down, some of that energy is released. That energy is going to be harnessed and converted into the form known as ATP, because that form of energy is something that the cell can actually utilize to do all the things that it needs to do. So if we look at a little more specific view of the equation, we oftentimes refer to glucose as the model molecule to follow through the pathway. Now, it doesn't mean that that's the only thing or the only fuel source that can be utilized, but it is the sort of the model or molecule that we follow. So glucose in the presence of oxygen, again, is results in carbon dioxide production, water, and the release of energy. Now, these reactions are all fall into a family of reactions known as redox reactions. These redox reactions are short for oxidation reduction reactions. And these reactions are defined by the loss of an electron, which is an oxidation, and the gain of an electron, which is a reduction. So sometimes students ask me, well, we're talking about energy, why are we jumping to talking about electrons? And the reason that we are talking about electrons is that electrons do in fact hold a great deal of energy. And so it is the transfer of these electrons that essentially is a transfer of energy that is occurring from one source to another. So redox reactions are really important because they allow for this electron transfer to take place, but for our purposes, that is a transfer of energy that is taking place with the movement of those electrons. So if you look at the summary equation down at the bottom of this slide, again, you see glucose and oxygen resulting in carbon dioxide and water. And again, what's missing from this is the production of energy. But glucose is undergoing an oxidation. It is losing electrons in this process. And oxygen is undergoing a reduction. It is the final electron acceptor in the overall pathway, and we'll highlight that again when we get to the end of the pathway. So questions about this sort of summary overall of cellular respiration before we look at a little more detail? No questions? Okay. All right, so before we again jump into a little more detail, I kind of want to highlight a couple of characteristics or components that are really important to cellular respiration. The first one up at the top is the term dehydrogenase, or plural as it's written here, dehydrogenases. Dehydrogenases are a family or class of enzymes that are involved in removing a pair of hydrogen atoms from a sugar or another fuel source. And the important thing here is that by removing these hydrogen atoms, excuse me, essentially what we are doing is removing electrons and protons. So this goes back to way back in the beginning of the semester when we talked about hydrogen atoms. We said that hydrogen atoms were a special type of atom and that they are made up of protons and they are made up of electrons. So by, whoops, sorry, go back, trigger finger here. This class of enzymes is going to remove hydrogen atoms. And again, by doing so, it's really removing protons and electrons. And it's those electrons that we're really after because those are the molecules or the components that contain the energy associated with them. So I highlighted in that last lecture several places in both the glycolytic pathway as well as the Krebs cycle where dehydrogenases come into play. Okay, give me just 1 minute here. All right, give me just a second here. Still figuring out everything here for me. All right, let me go back now. All right. Sorry, I'm really not great at doing two things at once. And so this is really testing me on doing multiple things at the same time. Okay, so. All right, I don't think this is what I wanted either. Give me just another minute here. Okay, so back to this particular slide with dehydrogenases on the title or the header. So dehydrogenases, the family of enzymes that are going to be involved in this electron transfer. The molecule that is going to be accepting these electrons is known as NAD. So that's right at the center of the slide. NAD is short for nicotinamide adenine dinucleotide. And this molecule is also a type of molecule known as an activated carrier. It's an activated carrier because it can pick up energy, in this case in the form of electrons. When it does so, it becomes NADH, like you see on the right-hand side of that equation. NADH now has the electrons associated with it. It has a great deal of energy associated with it. NADH can now take that energy, transfer it to reactions that require the energy, and by doing so, again, it is transferring the electrons. When it does so, it is converted back into NAD. So this is also a cyclic process like we discussed with ATP and ADP, in that NAD can pick up electrons. When it does, it's picked up the energy and it becomes NADH. NADH is the molecule that has the energy associated with it. It can then transfer that energy in the form of electrons to other reactions that require it. When it does so, it is converted back into NAD. So it is a cyclic process between NAD and NADH. NAD, NADH overall. And if you've looked at or listened to the previous lecture, you will see why generating NADH was so important in the processes of cellular respiration. Okay, so let's see. The last thing on this slide is really just to highlight that what cellular respiration is doing overall is producing ATP that the cell can utilize. It does that in one of two ways. One way is a direct synthesis known as substrate level phosphorylation. And here you have enzymes that are involved in transferring a phosphate group from one substrate to ADP, which is also another substrate, to form ATP. So this is a direct transfer of a phosphate group from one substrate to ADP to produce ATP, and this is referred to as substrate level phosphorylation. So this is direct synthesis of ATP. The other way that ATP is synthesized is by oxidative phosphorylation. Now, oxidative phosphorylation is an entirely different type of process in producing ATP, and it requires or utilizes these electron transfer reactions that occur, these redox reactions that occur, to then drive the synthesis of ATP. Okay, so questions about this particular slide before we move on? No, okay. All right, so cellular respiration is in fact divided up into 3 distinct metabolic stages. The first one is known as glycolysis, and glycolysis literally translates to the breakdown of glucose. Glyco refers to glucose or other carbohydrates. Lysis is a form of lyse, which means to break apart. So in glycolysis, we are breaking apart glucose in this series of reactions. Glycolysis does occur in the cytosol or the cytoplasm of the cell, and glucose enters the pathway. Pyruvate is produced. It is a 3-carbon molecule that is produced, so glucose is a 6-carbon molecule. At the end of glycolysis, we have 2 3-carbon molecules known as pyruvate. Pyruvate then can enter the next phase, which I'll get to in just a minute. There is direct synthesis of ATP via glycolysis by substrate level phosphorylation. In addition to that, there is some NADH that is produced. So remember, NADH has electrons associated with it, has a great deal of energy associated with it. And that energy is extracted from the breakdown of glucose in the glycolytic pathway. We next have, in the center of the slide, the Krebs cycle, also referred to as the citric acid cycle. So in your book, I think it's referred to as the citric acid cycle. Older terminology is referring to it as the Krebs cycle. Those are synonymous. Krebs just gives credit to the scientist who first elucidated the pathway. Citric acid cycle is appropriate because one of the components in the process is citric acid. So the pyruvate that was generated during glycolysis enters into the mitochondrial matrix, so the innermost part of the mitochondrion, and this is where the Krebs cycle takes place. The Krebs cycle does produce a small amount of ATP directly by substrate level phosphorylation. And there is also NADH produced. And as you see sort of at the top of the slide, there's also an additional type of molecule known as FADH2. This too is a type of activated carrier. FAD has less energy associated with it, can pick up electrons, and is converted into FADH2. And that is, again, a molecule that has energy associated with it. Okay, let me answer a question real quick. Cardi, can you go ahead and ask your question?
Do we have to memorize the Krebs cycle?
Not the specific steps in the cycle. So same thing with glycolysis, and I think I mentioned this in the voiceover version. You don't need to know every single step, but you do need to know the highlights of what's happening, and we'll summarize some of that in the next slide.
Okay, I just wanted to show you a few videos before we move to
another question? Uh-oh. We need to mute Cardi, I think. I'm gonna go ahead and mute her. There we go. If she comes back, she can ask a question. Okay, so then lastly on this slide is the third metabolic stage, which is known as the electron transport chain. The electron transport chain is where the most ATP is produced overall throughout the entire cellular respiratory pathway. And this is going to occur via oxidative phosphorylation. So all of that NADH and FADH2 produced in glycolysis and the Krebs cycle is then shuttled over to the electron transport chain. Those electrons that were picked up from the reactions we just talked about are donated or transferred to the electron transport chain. There are a series of redox reactions that take place, and in this case, this is where oxidative phosphorylation allows for the production of ATP. Okay, so questions about this particular slide? Just the overview of the 3 metabolic stages? No, no questions? Okay. All right, so this is an overview of glycolysis more specifically, and you see 2 different versions of basically the same thing. The figure on the left has a little more detail with the individual components in each step in glycolysis, and like I just mentioned, you don't need to know every single step. What I do want you to know and realize is more of what you see on the right-hand side of this slide, and that is what we consider or call the energy investment phase versus the energy payoff phase. So the first half of glycolysis is referred to as the investment phase. That's because ATP is required for some of the steps in the reaction. So in order for glycolysis to continue, we have to actually add energy into the system or the reactions. And then sort of at the halfway point, where the red line is, you can see below that is the energy payoff phase. This is the payoff phase. This is where ATP and NADH is being produced overall. And so it's referred to as the energy payoff phase. If you look at the very bottom of that figure on the right-hand side, you'll see a summary of what's happening in glycolysis. So this is probably the important parts to understand, that there's an investment phase where ATP is required. There's a payoff phase where ATP and NADH is produced. But if you look at again at the bottom is the net. So what we're seeing here is a summary of the reactions taking place. Glucose is required. Again, it's the model molecule that we follow. Glucose enters the pathway. So 1 glucose enters the pathway. 2 pyruvate are produced, as well as 2 water molecules. In addition, there's a requirement for 2 ADPs so that 2 ATPs can be produced. There's also a requirement for 2 NAD so that 2 NADH molecules can be produced. Now that's a net equation. If you look above that again, in the energy investment phase, we required 2 ATP. In the energy payoff phase, we synthesized 4. So if we synthesize 4, but we need 2, that's a net gain of 2 ATP. Okay, so questions about glycolysis in the summarized form. Okay, and Cardi, if you do have a question, you can go ahead and unmute yourself and ask the question, but I think you might have just left your hand raised. Okay, so no questions about glycolysis in general? Okay. All right, so then the next metabolic stage is the Krebs cycle, also known as the citric acid cycle. Now, the citric acid cycle, or the Krebs cycle, occurs within the mitochondrial matrix. So that's the innermost part of the mitochondrion. Just to review, every single mitochondrion has a double membrane system. There's an outer membrane as well as an inner membrane. And the inner membrane has extensive folding sort of back and forth and back and forth. That extensive folding provides surface area for all of the enzymes that we're going to get to in just a minute in the electron transport chain to interact with the membrane. So it just basically gives a platform for all of the enzymes required or necessary for the electron transport chain. Okay, so the pyruvate, excuse me, the pyruvate that was generated from glycolysis needs to enter into the mitochondrial matrix. Now, before it can do that, there is some conversion that occurs to pyruvate. So pyruvate is converted into a molecule known as acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, and acetyl-CoA can enter into the Krebs cycle. So this conversion takes place, which allows for the entry of acetyl-CoA into the mitochondrial matrix. But what you see there is that there is NADH that is produced in that process overall, as well as carbon dioxide release. So acetyl-CoA can enter into the Krebs cycle. And like I mentioned with the previous slide, in the last lecture, we had the steps outlined. You don't need to know every single step. What you do need to know is what's entering in to the pathway and what's being produced. So what you see here is in the cycle, so we're still on the left-hand side of this figure, you can see that NADH gets produced. You also see some ATP being produced directly by substrate level phosphorylation. And then you also see FADH2, that molecule that I mentioned just a little bit ago. So we can do a little bit of accounting at this point. For every 1 pyruvate that enters, 1 ATP is directly synthesized, 4 NADH are produced, 3 specifically in the Krebs cycle, 1 in that step converting pyruvate to acetyl-CoA, so that's a total of 4, and 1 FADH2. The next column over I have, though, is per glucose. So remember, when we talk about glycolysis, we started off with glucose entering the pathway, and glucose gets converted into 2 pyruvates. So this cyclic process happens twice for each pyruvate that is generated from glycolysis. So if we just double check the accounting for every glucose that has entered glycolysis in just this slide or this version of what we're looking at, 2 ATP are produced, 8 NADH, and 2 FADH2. All right, questions about this particular slide? No? No questions? Okay. All right, so the third metabolic stage is known as electron transport, or the electron transport chain. This occurs in with the series of proteins that are embedded in that inner mitochondrial membrane overall. What you see in this figure that's in front of you right now is the components that are part of the electron transport chain. Here too, I'm not going to ask you for any specific name of individual components of the electron transport chain, but what you should be really aware of is what's happening in this process. And what's happening in this process is the NADH and the FADH2 that was produced by glycolysis and the Krebs cycle, excuse me, transfer their electrons to the electron transport chain. Now, the electron transport chain is a series of redox reactions. Electrons are being transferred from one component to the other. And in doing so, there are 3 different locations that hopefully you can see with those red arrows that allow for hydrogen ions, or otherwise known as protons, to pass across the inner mitochondrial membrane. So those 3 red arrows indicate hydrogen ions or protons that pass through the membrane from one side to the other. So that energy in transferring electrons has been harnessed in a way to allow for those hydrogen ions to pass across the membrane. So this figure I think I like a little bit better just because it has the molecules sort of embedded in the membrane, but also gives you some idea of where they are located. So smack dab in the middle of this figure, you see the inner mitochondrial membrane. The components of the electron transport chain, most of them are embedded in that membrane. At the bottom, you can see NADH comes in, transfers its electrons to the first component of the pathway. And when it does so, that then allows for those electrons to be transferred from one component to the next. And as sort of a byproduct, if you want to call it that, the energy from transferring the electrons also allows for hydrogen ions or protons to get pumped across the membrane. So what we're doing by transferring those electrons is also simultaneously generating a concentration gradient with those hydrogen ions or protons. We are building up a high concentration of hydrogen ions in the inner membrane space compared to the mitochondrial matrix. By pumping them across, we're generating a high concentration of those hydrogen ions on that side of the membrane. FADH2 can also transfer its electrons to the electron transport chain. It does so at a slightly different location than NADH, but the effect is the same. Hydrogen ions get pumped across the membrane. Okay, so a little more accounting, and that is for every NADH that transfers electrons to the electron transport chain, 3 separate hydrogen ions are pumped across the membrane. Let me see if I can get my arrow function here working. There we go. So there's the first hydrogen ion pumped across, there's the second hydrogen ion pumped across, and there's the third hydrogen ion pumped across. So again, for every NADH that transfers electrons, 1, 2, 3 hydrogen ions get pumped across the membrane. Now, because FADH2 enters at a slightly different point in the pathway, only 2 hydrogen ions get pumped across. But we're still generating this concentration gradient of hydrogen ions in the inner membrane space. Higher concentration there compared to the mitochondrial matrix. Okay, so before I move on, because there's one last thing that's really important that we need to talk about, is there any questions in general about the electron transport chain and what's happening as a result of the electron transport chain? So Cardi, if you do in fact really have a question, go right ahead and unmute yourself and ask the question.
Can you hear me?
Yes, I can.
Okay. Does the kinase also play a role in the electron transport system?
The kinases?
Yeah.
No. So kinases, I actually wanted to talk about just a little bit ago, but I forgot to mention them this time. I mentioned them in the last lecture. Kinases are enzymes that do in fact transfer phosphates from ATP to a different substrate. So they are not playing a role in the electron transport chain. They are an important family of enzymes that remove phosphates from ATP, but they are not involved in the electron transport chain.
Oh, okay. Also, is our test on, is it on Tuesday after spring break?
I will have it open after Thursday, after our lecture on Thursday. So it'll be open, but it'll be open for an extended period of time.
Oh, okay. Thank you. All right. That's it. Thank you.
Okay.
Cardi B out.
Okay. So any other questions about electron transport chain before we get to like the really important part of this? Okay. All right. So hopefully what you have gathered from this is that we have generated this concentration gradient, a high concentration of hydrogen ions in the inner membrane space compared to the mitochondrial matrix. The most important part here then is this last molecule known as ATP synthase. Now, ATP synthase is a molecule that can allow for hydrogen ions to pass back through the membrane. And when it does so, can harness that energy of these hydrogen ions passing back through the membrane, going from that area of high concentration in the inner membrane space back over to the mitochondrial matrix where there's a lower concentration. It harnesses that energy to synthesize ATP. So this is the oxidative phosphorylation portion, in that ATP is being produced, but the way that it's being produced is due to those hydrogen ions coming back through the membrane from the inner membrane space across to the mitochondrial matrix, and that energy is harnessed to then produce ATP. This is where most of the ATP is produced in cellular respiration overall. Okay, so questions about that? Yeah, Jack, go ahead. If you can unmute yourself and then ask the question. No, do you need me to unmute you? Let's try that. Well, let's see, for some reason, I see there's a problem. You can't unmute yourself and I can't unmute you. I'm not sure what the problem is. So maybe you can type your question in the chat and, okay, got it taken care of. Never mind, we got it all squared away. All right, so any other questions about this slide before we move on? No other questions? Okay. All right, so what I'd like to talk about at this particular point is that this whole process does not require glucose alone. Glucose is in fact the model molecule that we follow throughout the entire process. But that is not the only molecule that energy can be harnessed from in order to produce ATP for the cell. When we think about what it is that we as humans consume, we don't consume glucose purely on its own. We typically have a diet that's composed of proteins, carbohydrates, and fats. I think the big joke now on social media is that we're all eating way too much lately with all of our quarantine snacks, and we're going to come out maybe 20 pounds heavier afterwards. Hopefully that's not the case. But again, our diets are not made up of glucose alone. They're made up of a combination of different components. And I really like this particular slide because it hopefully drives home the point that other components, other fuel sources can in fact be utilized to produce energy that the cell can utilize. So we'll start now at the middle because that's probably the easiest to understand, and that's looking at carbohydrates in general. So carbohydrates are polymers of monosaccharides linked one right after the other. Those can be broken down into simpler sugars. Some of those sugars can be converted into glucose and then enter glycolysis directly. So that's pretty straightforward, easy to understand example. Some of the sugars, however, can be converted into other components that can enter into the pathway at different locations. So even though we consume carbohydrates as a type of macromolecule, they can be broken down and can either enter directly into glycolysis or be converted into components that can shuttle in at different locations in the glycolytic pathway. Once the molecule is in the pathway continues on with all of the processes that we talked about. So again, for carbohydrates, pretty straightforward as far as how they can be utilized as fuel sources to produce ATP. If we look at proteins, however, proteins, a different class of macromolecule altogether. Proteins are polymers of amino acids, so the first step is going to have to be to break down those proteins into individual amino acids. Another really important step is the removal of the amine group from those amino acids, and it's a series of steps that occurs to do that, but that is important to remove the amine group. Once the amine group is left behind, what's left behind is a 2-carbon molecule. That 2-carbon molecule can be converted and enter into the pathway at glycolysis. It can also enter into the pathway where acetyl-CoA is. It can also be shuttled into different locations in the Krebs cycle. So proteins, we know, are a fuel source, but we might not have thought of how that really is providing us with fuel. And that is because they're broken down into individual amino acids. And then eventually those 2-carbon skeleton molecules can enter into different locations to allow for ATP production. Lastly, we have fats. And fats we oftentimes don't think about as being something that's good for us, but it turns out that they are, in fact, very high energy molecules. Fats are made up of glycerol and fatty acids. So if that's unfamiliar to you, you should probably go back to the early parts of the semester and review some of those macromolecule components that we discussed. But fats can be broken down into glycerol, which is a 3-carbon molecule, which can be converted and enter into the glycolytic pathway. Fats are long chains of hydrocarbons. Those hydrocarbons are broken down into 2 carbon units in a process known as beta oxidation. Beta oxidation makes these 2 carbon units, which eventually can be converted and enter into the pathway here at acetyl-CoA. So what you can see is that there's a lot of versatility with catabolism in general, breaking down large molecules to eventually be converted into components that can in fact enter at different stages to still produce some ATP from that fuel source. Okay, so questions about this particular slide? No questions? Okay. Okay, so the next thing that I want to talk just a little bit about is the process of fermentation. So fermentation is the topic of the lab for this week, and it was the topic of the lab before we had our little break. Fermentation is an alternative process that allows for the production of ATP in a cell under low oxygen conditions. So first, let's talk a little bit about what happens under normal conditions where oxygen is present. So back to the summary equation, we said that oxygen is a requirement for cellular respiration to take place. The oxygen itself, and I mentioned this really briefly at the beginning of the lecture, is the final electron acceptor. Let me go back a couple of slides because I think I want to just touch on something before we talk about fermentation in more detail. So let me go back just a few slides to this place right here. So hopefully you guys can all see that, what I'm highlighting with what's supposed to be my laser pointer. There's oxygen. So oxygen is the final electron acceptor. It sits at the very end of the electron transport chain and is the last one to accept electrons as they get transferred from one component to the next. The last component is oxygen, and it takes those electrons with the presence of hydrogen ions and produces water. So oxygen is required for this process to take place. If there is no oxygen present, then there's no final electron acceptor. So what happens when electrons get transferred is they back up because there's no last component to transfer them to. So again, oxygen is required for this process of cellular respiration. And that's this portion of the slide here. We're looking at an aerobic function. Aerobic meaning with oxygen or with the presence of oxygen. And we know that we go through glycolysis. Pyruvate gets converted into acetyl-CoA, enters into the Krebs cycle, and we get the electron transport chain for the production of ATP. All because oxygen sits at the very end of the pathway and is required to catch those last electrons from the electron transport chain. All right, so what happens if there's no oxygen present? If there is no oxygen present, that's referred to as an anaerobic situation. And in an anaerobic situation, pyruvate gets shuttled into an alternative pathway. This is an alternative pathway that allows for minimal amounts of ATP to be produced, very little ATP in comparison to the overall process of cellular respiration. But it is a mechanism that allows for some ATP to be produced so that the cells can in fact still survive if this is a temporary situation. If it's not temporary, then the cells will eventually run out of components required for fermentation and the process will stop and the cells will not survive. So again, it's a temporary fix, hopefully to get over the stress of not having oxygen for a short period of time, but eventually oxygen is going to be required to allow for the continuation of cellular respiration. All right, so there are actually two different mechanisms for fermentation. There's what's known as alcohol fermentation and lactic acid fermentation. And really, it just depends on the type of organism that we're discussing as to which one of the types of fermentation takes place. Yeast and bacteria, for example, can undergo the alcoholic fermentation pathway. There are also examples for lactic acid fermentation where bacteria, fungi can be utilized. This is in fact how a lot of cheeses are made through lactic acid fermentation. Human muscle cells also undergo lactic acid fermentation under times of temporary loss of oxygen. All right, so what's happening in these pathways is going to require a little bit of explaining. What you see up at the top, we'll look at alcohol fermentation first, and then we'll transition to lactic acid. But the mechanism is the same. Glucose is going to be converted into 2-pyruvate via glycolysis, and that is where the ATP that is getting produced does in fact get produced. We know that glycolysis requires NAD to allow for NADH production. If there is a lack of oxygen and the electron transport chain cannot continue, we will get a buildup of NADH in the system and no NAD. If there's no NAD, then glycolysis cannot continue. So what fermentation does is allow for the regeneration of NAD so that NAD can be utilized in glycolysis, most importantly to allow for this ATP production. So pyruvate gets converted into acetaldehyde, but most importantly is this step here again where NADH is converted back into NAD. And like I said, this is important because NAD is necessary for glycolysis to continue. And that pathway is exactly the same as far as functionality is concerned for lactic acid fermentation. The end molecule is different, but the process of taking NADH, restoring some NAD so that glycolysis can in fact continue is what's important from this mechanism. Okay, so questions about fermentation? No other questions about fermentation? Okay. give me just 1 second here. I think I unmuted you guys there for a minute. Okay, so let's see. Were there any questions about fermentation? No, no questions about fermentation? Okay. All right, so oftentimes I get asked, well, how much ATP does actually get produced overall? And the answer to that is it varies from textbook to textbook, depending upon how you want to do the accounting. So my counting is a little bit on the conservative side. It doesn't mean that the final number that we're going to come up with, that we're going to go over right now, is exactly the set in stone number. It is an estimate of the amount of ATP that gets produced. Okay, so keep that in mind. It's an estimate. It's not a set in stone value overall. All right, so what you're looking at in this modified table is the two metabolic stages, actually, glycolysis and the Krebs cycle. Then what we see is direct ATP synthesis, NADH production, and FADH production. So if we look at direct ATP production, glycolysis has a net of 2 ATP produced. Krebs cycle also has a net of 2 ATP produced. And then that gives us a subtotal of 4 ATP produced directly by substrate level phosphorylation. We also know that glycolysis results in the production of 2 NADH. The Krebs cycle results in the production of 8 NADH per every 1 glucose that entered the pathway, for a subtotal of 10 NADH. Now, go back to thinking about when we talked about the electron transport chain. With the electron transport chain, what we have is 3 hydrogen ions get pumped across the membrane, and those 3 hydrogen ions can eventually go back through ATP synthase one at a time to then allow for the production of ATP. So this is why I multiply by 3. For FADH2, 2 are produced in the Krebs cycle for every 1 glucose that entered the pathway at the beginning of glycolysis. And we said that FADH2 allows for 2 hydrogen ions to get pumped across the membrane. So that gives us a total of 4. So overall, again, a rough estimate of how many ATPs are produced for every 1 glucose that enters the pathway is a total of 38 ATPs. Okay, I see I have a couple of questions coming in the chat, so let me try and answer those for the group, and then I'll open it up for additional questions in just a minute. Okay, let's see. Okay, so I have a question that says, NADH and FADH2 is produced in the Krebs cycle, and then a small amount of ATP. So the answer is yes. Let me go back to that slide. Let's look at it here. So with the Krebs cycle, there is a total of 4 NADH. There's 1. And then there's 3 in the actual cycle. So this first one is not actually part of the Krebs cycle. It is the step in going from pyruvate to the mitochondrial matrix in acetyl-CoA. But then once the Krebs cycle is actually in place, 3 additional NADH are produced. So overall, everything in this red box, there's 4 NADH produced for every 1 pyruvate, 1 FADH2, and 1 ATP by direct synthesis. So I think that's what the question was, is that NADH and FADH2 are produced in the Krebs cycle, and then a small amount of ATP. So I think I've answered the question yes. And if I haven't, go ahead and ask me another question. Let's see. All right, another question is, there's no oxygen in the electron transport. Let's see. If there is no oxygen, the electron transport chain stops and little ATP is produced. So the question is, if there is no oxygen, the electron transport chain stops. The answer is yes. If there is no oxygen, the electron transport chain cannot continue. A small amount of ATP does get produced, but that is because the fermentation pathway kicks in. So hopefully that answers the question as well. All right, are there any other questions about cellular respiration overall? If you want to use the raise hand function, you can, and you can ask it by unmuting yourself, but I also have the chat function and I can address the questions there as well. Okay, so this one's not necessarily related to the content of the lecture, but for exam 3, we should only do the objective questions for cellular respiration and photosynthesis and not cell division and genetics. The answer is yes. I have not gone back through and looked and updated the objective questions, but I will try and do that today and get them updated. But the short answer is yes, we have not covered cell division and genetics yet, so that will actually go on the final. What will be on this exam after this week will be the little bit of review that I did post for the material that we covered before our break, cellular respiration, fermentation, and photosynthesis will be on exam 3. So hopefully I've answered that question then. Okay, are there any other questions? Okay, so question is, can I go over why I multiplied the subtotals again? Absolutely, I can go back. Let me go to that slide. Give me just 1 second. Okay, so I'm just gonna uncover it all so we can look at it. All right, so I'm gonna go over this this one more time just to make sure that it's sort of clear as to what's going on. So looking at direct ATP synthesis, we know that there is substrate level phosphorylation that produces 2 ATP in glycolysis and 2 ATP in Krebs cycle. And this is again considering 1 glucose that enters the pathway. So that gives us a subtotal of 4 ATP that are directly synthesized in those processes. Okay. For NADH, we know that in glycolysis, 2 NADH are produced, 8 are produced in the Krebs cycle. Again, this is for 1 glucose entering the pathway at the beginning, going through both glycolysis and Krebs, for a subtotal of 10. And then the multiplication here is by 3. The reason for that is that for every NADH that transfers electrons to the electron transport chain, 3 hydrogen ions get pumped across the membrane to generate that concentration gradient. If all 3 of those go back through ATP synthase, that's how many ATPs we would synthesize. This is why it's a rough estimate, because it's not necessarily a 1-to-1, but I like to make the calculations easy and just make the assumption that it is a 1-to-1. So again, this multiply by 3 is because 3 hydrogen ions get pumped across to generate the concentration gradient. Each one of those 3 can go back through to synthesize ATP by ATP synthase. And so then the multiply by 2 for FADH, is coming into play because FADH2 enters or donates its electrons at a different point in the pathway. So only 2 hydrogen ions get pumped across the membrane. And if only 2 get pumped across the membrane, only those 2 can come back through to produce ATP. So does that answer the question, I hope, Nika, for you? Okay, great. Okay, any other questions? No, no other questions. Raise hand function if you have another question. No other questions? No? Okay, well, if there are no other questions, we'll go ahead and end our little Zoom session here. Like I mentioned, yes, go ahead, question. Yes.
No, I just wanted to say thank you for the lecture. I just want everyone to stay home, stay safe from, you know, the virus. And until next time, okay?
Yes.
I do what they say I can't. They call me Cardi, Barty, Banging, Bobby, Mommy, Habitat.
Great. Okay, any other questions? No? Okay, so like I said, we'll go ahead and stop our Zoom session here. Hopefully after spring break, I will have my video docking system set up where I can actually write and you can see what I'm writing. All of this is obviously taking some time to get set up, and turns out that equipment is a little bit hard to come by these days because, you know, every campus in the United States is doing the same thing. So hopefully we'll have that after spring break so that some of our Zoom lectures, I might actually be able to write, you know, write on a piece of paper so you can see some of that as well. But I am, again, always open to questions. If you have questions, please shoot me an email. You can also use the discussion function in our class Blackboard. And if you need some one-on-one time, I'm happy to set up a Zoom session with you as well.
Oh, yeah.
So if you are interested in doing any of that, we can do that as well. All right, no other questions? Okay, guys, definitely stay safe, stay home, and I hope to see you all again really, really soon. Bye.
Thank you.
Bye. You're welcome.
Love you.
Bye.